Alrighty. Well, welcome everybody to our NGS tech session. This is a long-standing tradition of this meeting. We've had a lot of fun over the years with this, and we've got two of our mainstays, both sponsors again this year and have been for many years. We have Illumina and PAC Bio to tell us about their technology. So one reminder, by the way, you can post questions or in the question and answer box at the bottom of your Zoom screen, bottom right corner of your Zoom screen. And again, please visit the sponsor sites and we'll go ahead and get started. So we're going to start with Illumina. So we're going to do two 20-minute talks. With Illumina going first, and then PAC Bio, and then we'll have about 20 minutes or so, 20 minutes or so of questions. So we're going to start out with Illumina and Haley Fiske, who's the director of marketing. Haley's been in the genomics industry for over 30 years at companies such as Applied Biosystems, Molecular Dynamics, Swift Biosciences, and Illumina. And currently at Illumina, he manages a team of DNA sequencing specialists. Haley's an old pro at this panel. He's done it for many years off and on. So today, he's going to talk about unlocking the power of genomics for pathogen detection and surveillance. So go ahead and take it away, Haley.
Thank you, Bob. And I want to thank the rest of the organizing committee for inviting me here today. Now let's see if I can share my screen properly. Here we go. Can you see that? All right. Yep, so I'm here today to talk about unlocking the power of genomics for pathogen detection and surveillance. In my talk, I designed it in the sense that I just want to get everybody thinking, and hopefully it leads to more discussion afterwards. There is a paper it's a review paper that was published in 2017, and it came out of Dr. Fauci's lab, and there's these two graphics that struck me, and one of them shows, and this was shown to Congress in 1984, and Fauci pointed out that there was one emerging threat, HIV, and then 2017, after some 30 years of following threats or outbreaks, there was more than 30 globally. Now, I know we've all been in the various stages of the pandemic for the last, say, 18 months. But what I found interesting about this paper is if you look back, there are lessons that were learned over these 30 years. And the lessons that are learned, and even in the last panel, I was hearing, some of these same responses. It's that global surveillance, what you need a global surveillance to detect early outbreaks, having transparency and communication in response to the outbreaks, different labs communicating with each other, and then the ability to incorporate, the infrastructure of the private companies internally and internationally in response, and then to, conduct basic research and clinical research associated with the outbreaks in like a coordinated and collaborative manner. And then also involving, and I think this is really important, with respect to how the population is responding to the pandemic, it's involvement of the afflicted communities in policy decisions. And then pursuit of technology platforms that you can build vaccines with, or diagnostics as therapeutics. And of course, flexible funding. And so if you see this was written in 2017 and you kind of look back over the past couple of years, and it's like we knew what to do, and all of us that have been coming to this meeting knew what to do. And I think it was more of the pandemic became so large and global that others had to come up to speed on what needed to happen. Now, and this is a kind of a shout out to the past SAF meetings. We knew it was coming. Like every time I'd go to a meeting, the organizers would have 1 or 2 talks about some emerging pathogen or some outbreak. I'd come home and my wife would go, "Oh, how was the conference?" And I'd go, "Well, I definitely know now what not to eat and where not to travel." And so I want to say that we knew it was coming. Hollywood knew it was coming. And I think most of us, when we were in the midst of the pandemic, we thought initially it could be beaten. And I'd just like to say that, 5 waves and almost 5 million people later, I don't think any of us think that at this point it can be beaten. And, I think we're gonna likely be, who knows how fast it's going to taper down. I mean, we still have issues with global vaccination. We have issues in the United States with people unwilling to get vaccinated. But at the same time, I think we have to start looking forward. And in terms of looking forward, I believe, and I think a lot of you believe, that COVID will become endemic. And for me, there's 3 clear signs. And I think this is what got us at the beginning, is that there's asymptomatic transmission. So unless we were going to test every single person globally, every 2 or 3 days or once a week, we wouldn't be able to find the asymptomatic people and have them stay at home. So we're not going to be able to solve this problem. Then there's the whole aspect of infection breakthrough. So you're going to have individuals that can still get infected and spread the disease. And we've been learning since the beginning of the pandemic that we're starting to see other species that contract coronavirus, and they're going to become reservoirs for a virus. So in the future, there will be outbreaks from wild animals or animals in markets that are eaten. In terms of what I would say Illumina has to offer and what the community can do with DNA sequencing products, I don't think a lot changes moving from what we were doing pre-pandemic to what we're doing, what we're going to do when it's endemic. I just think it's going to be larger, bigger networks, a lot more individuals involved in surveillance, but we're still going to have to do the same thing. And that's identifying and characterizing, understanding the mechanisms, understanding the transmission between animals and humans and animals and other animals. We're going to have to remain vigilant in terms of epidemiology and surveillance. That's, understanding where the reservoirs are and how they spread. And then we're also going to have to keep vigilant with respect to vaccine development. So if the spike protein changes and then the vaccine isn't effective anymore, a new vaccine or a modified vaccine is going to have to be invented, developed. So in terms of Illumina, we have a number of microbiology workflow solutions, and depending on the surveillance need or the type of organism or the specific organism, we have a number of, I would say, different workflow solutions. Now, with respect to moving forward in terms of an endemic disease, it is likely going to be a broad, my view is initially you'd want to use a broad targeted panel, or you might want to do some type of microbial whole genome solution. But there's a way to think about it, and that it depends on the target organism. Do you know what it is? Do you suspect what it is, or is it unknown? Now, if it's known, you want to use a targeted amplicon solution, and you don't always have to do this, but it's typically cheaper and faster. Now, if you suspect it, then you might want to use a custom enrichment solution, something with baits that you can pull out regions that you wouldn't be able to amplify with a primer. And then if it's unknown, you want to go with shotgun metagenomics. But typically the shotgun metagenomics piece is more costly from a library perspective, from a sequencing perspective, and from a data analysis perspective. Now, with respect to surveillance and monitoring, when it moves to the endemic phase, I would say that we have a panel, and we call it the respiratory viral oligopanel, that this could perhaps be one of the main tools that would be used. And it covers, the panel has baits for greater than 40 viruses. It's cost effective and sensitive, more sensitive than shotgun metagenomics. And if there's a mutation, say a deletion or a SNP below a primer for a PCR type panel, the enrichment panel will still work. Now, it also has a very simple workflow, and it uses what we call Illumina RNA prep with enrichment library prep protocol. The kit comes with a cDNA synthesis piece. So you take your RNA, you make cDNA, and then you add your adapters by tagmentation. And then there's a hybridization step. You complete the libraries, you quant, normalize them, and then you load them on the sequencer, and they take, it takes less than 9 hours turnaround time for the whole process. Now, sorry. Now, if you look at the total workflow with the RVOP panel, you're looking at 9 hours of library prep, and then depending on what instrument you have, whether it's a mini-seq, MiSeq, or NextSeq, you're looking at between 13 and 18 hours runtime. So roughly within a day, you can get, go from sample to answer. We also have informatics with the Dragon pathogen detection, and you can go from sample to data analysis all the way through to the report. Now, in terms of this panel, so there's the whole aspect of post-pandemic. If an individual has symptoms of, respiratory symptoms where they have a flu, a runny nose, they may want to also know, if you don't have SARS-CoV, you might want to know, well, what do I have? And so this panel covers human respiratory viruses, such as, it covers the other 4 common coronaviruses, covers adenoviruses, parainfluenza, influenza A, influenza B, it covers rhinoviruses, and it covers the Bacilla viruses. Now, this data here, I just wanted to show you in terms of this panel gives even coverage across the SARS-CoV-2 genome. And that's important because you don't want any sequences while you're sequencing to drop out. But what's also important about this panel is you can detect as low as 1 copy, and it has a large dynamic range up to a million copies. I mean, you probably detect more than 1 million copies, but you'd be wasting sequence. Now, in terms of monitoring, this data is from a publication that shows the use. It's a case study that shows the use of the RVOP panel in detecting SARS-CoV-2 in sewer samples. The samples were collected in Northern California in the East Bay. The research was done at Berkeley, Stanford, and Illumina. That's where the researchers are from. And you can see here in A, this is virus, or this is library that hasn't been enriched. And so you can see this blue is tomato viruses. And for those of you who may not be familiar with the analysis of sewage for viruses, there's a lot of plant viruses in the sewage, and these plant viruses were initially known to be there and are used as controls because there's the whole aspect of measuring from we'll say the point of the fecal matters entering the sewer to when it gets to wherever you're going to sample it, whether it's, say a manhole where a building is, or right before it goes into the sewage plant. These viruses were known and could be used as controls to try and figure out how much viral load was in this, at the start, and then how much, a day later or 2 days later, or with dilution from water coming in from rainwater or from other areas. But anyway, what this data shows is that when you look at total virus, you will see some tomato viruses, and then here B is with enrichment, and all you see here is coronaviruses and baculoviruses. And then also, this is for different dates from Berkeley, Oakland, and Marin, and you can see the unenriched, and you see tomato, pepper, and cucumber viruses. And then here you can see with the enrichment. Now, I also want to point out that with this kit, you're able to look at using next strain. You're able to see, or the researchers were able to see, where there were strains that were, in Alameda, but found nowhere else in the world, or in California, or in the US, or globally, or this gray here shows strains that had not been seen in California yet, and it was first picked up in the sewage. And then you can also see where here you can detect specific SNPs in different locations where the samples were collected. All right, in terms of the conclusion of this study, it just shows that you can actively sequence wastewater and get strain level genotypes. And another key advantage is, since it's enrichment, even if there's a SNP or a deletion under a primer, you're still going to pick up that sequence. And then you'll be able to, with a panel like this, you'll be able to look at viral distribution within communities and understand transmission. So in the future, when it's endemic, you'll be able to sort out like where it's coming from, and then figure out methods to stop the regional outbreak. Now, I also want to point out, and this data is from earlier this year, that there was a case in the Netherlands where there's transmission of SARS-CoV-2 on a mink farm, and it went from the humans to the minks back to the humans. And they have phylogenetic trees within this paper of the different minks that were infected and the different humans that were infected. And it occurred across 6 farms within the Netherlands. Now, after this paper was published, and this was there's a review article that you can go to and read about the infections in mink farms, but after it was published, then country by country, they started sampling mink farms, and the minks have COVID-2 infections pretty much everywhere. And this paper points out that, with respect to, so now minks are reservoirs, and then minks can likely infect other animals that are closely related, and humans can infect other animals. We're going to have numerous reservoirs, zoonotic reservoirs, likely in the future of COVID-2. And so even though as the human population post-pandemic, the infection rates drop, we're going to very likely have outbreaks from various animals. And with that in mind, there's recent publications, and I don't know if anybody's got any sequencing data from deer, but there's deer in the Midwest where 30% of the deer have antibodies. There's also been infections with dogs and cats. And then in all the zoos, they found infections, first in the Bronx Zoo, the animals were sick. And then in the Oakland Zoo, proactively, they took 48 different species of animals and they gave them vaccines. And it was hyenas, chimps, mountain lions, tigers, river otters, a lot of different species that they thought might get sick. So in terms of looking forward, in terms of monitoring outbreaks when the virus is endemic, a whole host of species are going to need to be looked at. Now, this is my pitch for today, and my pitch is this. So we're in the pandemic now, and it's going to start to wane. We need to get ready for the when the virus becomes endemic, and there's money that's been set aside, and there's going to be, you know, grants. And so I just wanted to make you guys aware that APHIS, and here's the URL below. There's going to be $300 million that's going to be available. You can go online, you can read about it, but essentially it's about addressing longstanding needs to strengthen our ability for emerging zoonotic diseases. They want to develop a framework. There's going to be, you know, strengthen our ability to detect in the future. And, you know, if you think about in terms of zoonotic disease, I think somewhere close to 70% of pandemics and outbreaks are initially animal born. You know, anthrax with livestock, seals brought tuberculosis to America, civets were SARS-1, bats, it was Nipah, Marburg, Ebola, and COVID-19, and the plague was rats and fleas. So my pitch today is you should, if you are working in a system or you want to work in a system, money's going to be available. Please apply for those grants. And when you get the money, Illumina has the tools to help you do the work. And with that, I just want to remind you that applying genomics and infectious disease and public management, you can do it all. The host genetics, host immunity response, pathogen ID, pathogen evolution, pathogen tracing, antimicrobial, I can't even say it, antimicrobial resistance, and drug and vaccine development. So thank you.
Great. Thanks very much, Haley. Good stuff. I'll hand it over to Donna and let her take it from here.
Sure. Okay, so our next speaker is from PAC Bio, and it's Dr. Meredith Ashby, and she's been with PAC Bio since 2009 and has worked in R&D on both sequencing optimizations and on bioinformatic applications development. She's now focused on facilitating the use of the PAC Bio long read sequencing to solve and to understand the incredibly diverse range of microbes that live in, on, and around us and on the systems that we depend on. Dr. Ashby's talk is entitled HiFi Sequencing for the COVID Era, High Resolution Pathogen Surveillance.
Okay. All right, and I just want to make sure y'all can see my screen, okay?
Yes, we can.
All right, great. All right, so thank you for that wonderful introduction. I wanted to start by introducing PAC Bio technology and note that the key differentiator for PAC Bio sequencing is our HiFi reads and the fact that they are both long and highly accurate. And that really makes us unique. And when you ask how accurate, you know, the answer is very accurate. We in fact have the highest accuracy of all platforms that are on the market today. And this is data that was released by the NIST study that looked at reference genome sequencing for human reference samples. So you don't have to take our word for it. And I also would just note that I probably am stating the obvious to this audience because a lot of you have been in fact, intimately involved with demonstrating the utility of HiFi reads for transforming genomics and generating more accurate and more contiguous genome assemblies over the last decade. So what I'm going to talk to you about today is maybe something that has not been the focus of many folks in this audience until the last year or so, and to talk about how HiFi sequencing is useful in the context of not just large genome assembly, but infectious disease. And so I'm going to touch on a couple different sequencing areas, starting with viral pathogens. So the first thing I want to note is that PacBio is coming out with a fit-for-purpose HiFi viral SARS-CoV-2 kit for doing surveillance of the SARS-CoV-2 genome. And some of the interesting things or differentiating qualities of this kit is that, first of all, it's a very simple workflow. It's an add-only enrichment procedure, which makes it super easy to use and simpler, less prone to errors than other amplicon-based technologies. This is not a traditional amplicon-based kit. And so stay tuned to get more details about exactly how the kit works. But the bottom line is that the method that we're using introduces greater robustness to the introduction of variants, because we are not dependent upon having just a single amplicon or maybe two amplicons at best covering each region of the genome. It's also easily scalable, anywhere from 22 to 384 samples per run on the Sequel II or 2E systems. And results can be had in 24 hours with automation, with only 3 hours of hands-on time. So that's all the detailed stuff. And now I want to get back more into the science and hopefully explain to you why HiFi sequencing is such an interesting proposition for pathogen surveillance. So the first question that I think a lot of people think is, when it comes to viral sequencing, is why do such small genomes need long-read sequencing at all? And the answer is because in many cases, viruses, once they infect the host, they have a relatively sloppy manner of replication. And so while you are infected by sometimes just one or a small number of virions, these can replicate and quickly develop into a cloud of only of closely related but distinct genomes that exist in your body. And these are called quasi-species. And this is where long reads are really useful, because instead of sequencing just the average of what's going on in the host, you can actually get a high resolution picture of all the quasi-species that are present and look at the relative abundance of these. And that can be really useful for detecting, understanding how variants of concern arise. How does drug resistance evolve and how common is it? How is immune escape happening and how frequently, and what are the mechanisms of it? And answers to those questions are very useful for making public health decisions, including the development of vaccine boosters. So here's an example of a researcher who used the long-read capability of PAC Bio sequencing in order to address some of these questions with SARS-CoV-2. And I'll say that this is an application that is different from the high-volume surveillance kit that we're offering. This is, again, using a longer amplicon in order to do that high resolution and of quasi-species and minor variants. So this work was done by Dr. Eli Boretz at NIAID, and what he did was develop an amplicon, which is a 6.1 kb amplicon that includes the spike protein, but also the E and M genes, and that's really where all the action is happening in terms of the immune system interfacing and interacting with the virus to clear it from your system. And what they found is that when they tracked viral load and sequence variants over time in one patient, that there was a lot of dynamics that were revealed when you were able to get this highly resolved view. So you'll see in the case of this patient, the viral load began very high when this patient was hospitalized, and then over time was starting to come down. But then around day 15, in gray and black, there was a resurgence of the viral load, and then that was coinciding with the appearance of variant sequences in the SARS-CoV-2 immune system, right? And so by tracking all of these things at the same time, they were able to really get a window into how natural selection is happening under host immune system pressure. So when they delved more deeply into where the mutations were occurring, you can see that they occurred predominantly in this NTD domain. And this NTD domain is interesting because it aligns with an epitope, which is known to interact with antibodies that have been flagged as being important for clearing the virus. from people's immune systems, right? And so this really is a key example of viral evolution happening under natural selection. And so applying this type of assay to more patients and more detail can start to give a window into questions like how do variants of concern arise, and also what should we be thinking about in terms of booster shots and booster shot development. So another virus, which is of tremendous public health concern, is influenza. So influenza is the only other virus that has already caused a global pandemic. And I think that the experience with COVID-19 has shown that it would behoove us to do better and more in-depth surveillance of influenza moving forward, now that we have seen the importance of generating information early. And in the context of influenza, some of the questions that I think will be interesting to put more attention to moving forward is about variants of concern and the amount of mutations. Flu, unlike COVID-19, mutates pretty rapidly, and such that mutations are even known to accumulate during the process of vaccine manufacture. And the current assays that we have for monitoring and surveilling influenza really provide only limited information about how often these variants are arising. So here's an example that was of work done at the University of Washington showing how more highly resolved information can be useful in understanding influenza. So in this case, they did single cell sequencing. They had an in vitro assay where they infected cells with influenza, and then they did single cell sequencing of those cells and looked at the diversity of the influvirus within each one of those cells. So as you move down the rows here, each row is a different cell. And then as you move across the columns, each column is a different one of the 8 genes that are present in the influenza genome. Anything that's in dark blue means there was more than one viral variant present in that cell. And then the other colors denote what types of variants were present. So yellow is deletions, and anywhere there's an orange dot or circle, that means there's an SNV that was either synonymous or non-synonymous. And so what the researchers saw is that after just 10 hours post-infection, only about 1/3 of the cells contained wild-type influenza. The rest had already replicated to form part of that cloud of quasi-species that I was telling you about. And these specific flu mutations that were observed were shown to make a difference both in terms of viral load in any given cell and also the triggering the expression of innate immune genes. And so this really gives a window into the kind of information that can be had with a high resolution approach to virus surveillance. So a second topic of interest for infectious disease is, of course, bacterial whole genome sequencing. So again, with what I hope will be an exciting product update to this audience, microbial whole genome sequencing application is migrating to HiFi. And so that application will be released in November. And some additional benefits that are going to come along with that is that all the sample prep reagents will now be in a single kit. We can do more microbes in a single run, so up to 96, which is improved from 48. And because we're using HiFi, we can also shear down to smaller insert sizes, which decreases the runtime for sequencing, but also gives us access to more of the smaller plasmids that are present in the sample, which in the past may have been filtered out of the library in order to focus on longer fragments to get that high quality assembly. The overall library prep has also been improved to make automation simpler and reduce hands-on time. And of course, there's the same dedicated microbial assembly pipeline that we have always had with all those bells and whistles, but now with HiFi reads doing the work behind the scenes. Okay, so antimicrobial resistance before 2019 was really the driving concerns of public health laboratories here in the US and abroad as well. Antimicrobial resistance is a huge problem for both human and animal health. It's a problem in our hospitals. It's a problem in our food systems. As we all start to move through the world in a more normal manner again, we expect antimicrobial resistance to resurge as a problem. And with the new investments that are now available for public health monitoring, We think that there's going to be a focus in bringing all the tools that were developed and brought online in our public health systems to bring those and make those available for this longstanding public health threat. And we are hopeful that that is going to lead more people to appreciate the more in-depth information that PAC Bio can bring as it becomes more fully integrated into public health labs. So PAC Bio is already playing a role in a number of public health systems. And here's one example of a network of Boston area hospitals that uses PAC Bio as one part of a pipeline to identify infectious disease that's being transmitted within the hospital. So in this case, this pipeline is very focused on identifying carbapenem-resistant Enterobacteriaceae, say that 3 times fast. And currently, most of the sequencing is done with the short read platforms, but anything that is new is flagged for long read sequencing with PAC Bio, right? And so this is the emergence of antimicrobial resistance. Again, it respects no boundaries, just as we saw with COVID-19. And in North America, there are some types of beta-lactamase carbapenem resistance that are already common, but there are others that really are only appearing outside the US. And there's a huge focus on monitoring whether these are entering the US and in what context, and whether they have achieved a community spread. So as part of this Boston area pipeline, they've actually identified a number of NDM resistance classes that are not yet commonly spread in the US. And so they wanted to understand what vectors were involved. And so the first lesson was that by doing that in detailed typing is that the strains are currently entering the health system sporadically instead of being spread within the hospital system, which is of course good news. So the thing that PAC Bio is able to add to the surveillance system is that high-level resolved view of how AMR is spreading in bacteria and moving through the system. So what you see here is this NDM common region, and this is an assemblage of genes flanked by transposons that enables it to spread through plasmids. So here are 3 different plasmid backbones where this same cassette is found, and also to be integrated into chromosomes. So this last example is a chromosome. And the thing that's super interesting about tracking through all of those different plasmid backbones is that antibiotic resistance, or this cassette, does not spread equally, or is not equally mobilized in all different contexts. So the place, whether it's in a specific plasmid or in a chromosome, has a big impact on whether resistance can be spread from one clade of bacteria into another. And so here you see across the x-axis a range of different possible hosts, right, that are replicated once for each different type of potential donor plasmid or chromosome. And what you'll see is that in blue, this plasmid background is a broad range host. So when resistance gets into this type of plasmid, it can easily spread through E. coli, E. cloacae, and other types of bacteria that are found in hospital systems. And in contrast, when these cassettes appear in other types of backbones, for example, this one here in gray or in red, that is inherently less worrisome because they are not as able to spread and disperse across many bacterial clades. And so the bottom line is that upgrading and expanding reference databases for antimicrobial resistance with higher quality genome assemblies is going to give public health professionals a more complete and less filtered view of AMR. And with this additional level of resolution and understanding, we can understand, where the real threats are coming from and how urgent they are and what actions need to be taken to protect the public health. So with my remaining time, I want to touch briefly on a third, more forward-looking application for public health, and that's metagenomics. So metagenomics can involve a range of different applications, full-length 16S sequencing, but also metagenome profiling, where you're really focused on enumerating and understanding the functions that are present in a community. And then finally, metagenome assembly, where the idea is to generate high quality references that can be then used for either 16S sequencing or metagenome profiling. So if I was going to give this talk to public health lab professionals, I would probably focus mostly on full-length 16S. But I know that the folks at SFAF are really excited about complex genome assembly. So I'm going to instead talk to you about some exciting results we've been getting with metagenome assembly using HiFi reads. So the bottom line is that HiFi reads are really uniquely well suited to metagenome assembly. And the reason is that metagenomes can contain very closely related strains, right? And a difference of only 3% is a common cutoff between defining what is a distinct species. And so long reads are certainly very helpful in overcoming assembly complexity. But the challenge is, if you have noisy long reads, the first thing that happens in the assembly process is to do error correction by mapping similar reads one against the other and washing out the errors in that context. And then those error corrected reads are put into the assembly pipeline. But in the context of metagenomes, what can happen is that you're basically, you can erase the true differences between closely related species that you need in order to come up with correct assemblies of all of the substrains that are present in that sample. And so you end up with things like species averaging or missing low abundance species, because those low abundance species are seen as errors. And you can also end up with these Franken assemblies, right? Assemblies that have parts from one strain and parts from another strain that don't actually exist in nature. The beauty of HiFi reads is that there is no error correction step. The HiFi reads go directly into assembly, and so you can avoid a lot of those consequences. And so you don't have to believe me. You can download some data. You can go to our website and find this data download and see where we have done sequencing of 4 different samples. These are averaged samples from the Bio Collective. 2 are from pooled vegetarian donors, and 2 are from pooled omnivore diet donors. And so unlike mock community samples, which are generally very simple, these are high complexity, but because they are pooled samples, they have like 1 million different aliquots that you can go to do your own sequencing, or you could just download our data and have a look at that. So here's some high-level results on metagenome assembly. So you can see that with just 1 SMRT Cell of sequencing, you can get up to 70 high quality mags per SMRT Cell of data. And that a lot of these actually appear in single contig mags. And so for those of you who are familiar with metagenome assembly graphs, here's an example of what that looks like. So you see we haven't entirely solved the problem of metagenome assembly. So there's lots of great work for you all to still do, but we are getting a lot of closed single contig genomes. And in terms of HiFi, this is a specific example that I like to pull out. So from this example, we have from this one donor, we have 2 strains, and both of these map to the closest species, Clostridium CAG217, right? So they map to the same species. They have highly similar genome size, but you can see that they have different coverage levels, which would seem to indicate that they are in fact separate species, even though and both are highly complete, right? And so if you have a look at the assembly graph, you can see that these 2 species correspond to these little blips on the assembly graph. And if you align them one against the other, you can see that they have an average nucleotide identity that's very, very high. But despite this, HiFi reads can assemble them cleanly into the distinct strains that they really are. So to summarize, PAC Bio is a powerful tool for infectious disease and public health, and we continue to push through longstanding barriers with genome assembly, including in the microscopic world. And our unique combination of long read length and high accuracy offers unique advantages, whether you're looking at isolates or complex samples like viral samples or metagenome samples. And that our technology can reveal many aspects of infectious disease that are invisible to NGS, but which are consequential for public health decisions. Thank you so much.
Thanks, Meredith. We will now open up questions for both Haley and Meredith. And please post the questions in the Q&A. It should be at the bottom of your screen, the right-hand side. And we already have a number of questions posted. So I think I'll just start and go down the list. Bob? Okay.
Sure.
So the first one I have is for Meredith, and that is what aspects of the HiFi sequencing pipeline, the long-range RT, PCR, and analysis software do we need to invest and for effort in order to differentiate quasi-species and low-level sequencing noise?
Yeah, so the paper that I referred to by Dr. Boritz and others at NIAID really included some innovative sample prep techniques to start to address this very real concern. PCR introduces error, and all sequencing technologies also have error. One of the things they did was of course to integrate UMIs on the ends of all the data. And they had also some specific algorithms, which I'm sure you will understand more than I did, to address things like PCR and sequencing error. The one thing that they flagged as being of concern is that the first step with any of these viral sequencing is the RT step, and RT enzymes do bring in quite a lot of error. And so that is an ongoing challenge.
Okay. So for One Health approach, how do you feel, how do you see your platforms begin to be used most effectively to both surveil human populations and zoonotic boundaries, while also working towards more effective use of NGS for clinical diagnostic applications? And I guess that's going to both, and whoever wants to take the first stab at this.
I can take the first stab. Based on, I would say, the maturity of our platform, we just have a lot more applications in library prep, and we can cover the whole gambit. And also, I want to chime in about our array offering. Like, there's been publications using arrays for you can get a methyl signature from a patient, and depending on that signature, it will you can predict whether the patient or how badly the patient will respond to the infection, and whether in terms of hospitalization, whether they'll die or not die. And so I think, with respect to our platform and One Health, you can cover the viral piece, you can cover in terms of, viral identification, strain typing. We've got a large RNA-seq offering. So in terms of any of the other organisms, in terms of how the virus affects gene expression, and then we have the whole genome sequencing piece. And depending on what type of lab you are, whether it's a small lab or I want to say a large scale, more industrial lab, we have a sequencer to meet your needs. So, 5 different platforms. And so if you're very high throughput, we have something for you. And if you're a very small health lab, we also have something for you.
Yeah, so at PAC Bio, I'm sure many of you are aware there have been a lot of changes at PAC Bio over the last year and a half. And some of that has included a strong pivot to look at infectious disease, but there's also been a strong pivot to make our platforms more accessible to a broader range of customers who may have struggled in the past with the high CapEx cost of our platform. And so I would say that from PAC Bio, what you can expect is more efforts in that area to make our technology more accessible. In the public infectious disease area, we are coming out with 2 new kits just in November to specifically address the needs of these customers. And I think that you can see us continuing to make investments in those areas. And we also have some special programs to get Sequel II instruments into public health labs and into metagenomics labs at special pricing, recognizing that some of the constraints about cost work a little bit differently in that context. And so I think that, keep an eye on PAC Bio. We are starting to pivot to really start to address some of these pressing public health concerns.
Great. All right, next question. What's the best way or point of contact for folks to work with your companies to help develop third-party assays on your platforms? Everything from assays, bioinformatics solutions, or really any part of end-to-end solution for sample in and answer out. Meredith, you want to start on this one?
Yeah, so you can email me and I'll put you in touch with the right person. At PAC Bio, all of our email addresses are really easy to figure out. It's first initial and last name at pacb.com. So if you want to, I can connect you with folks in any application area if you're interested in developing those kind of solutions, and we can see if it's an area where our interests align.
And I'd like to say the same. My email address is hfiske, HFISKE @illumina.com. But also, I supervise a team of sequencing specialists, and we're very technologically focused. And within wherever you reside, and if you're offshore, just send me an email. But if you're in the US, wherever you reside, talk to your sales rep, get the sequencing specialist name, and you can discuss with them about the technology and what you're trying to work on. And then we can feed you to the right person in-house, because we have a number of product management and marketing people with respect to library prep, and we can figure it all out.
Okay, I think this one is for Meredith, but Haley, if it applies to your platform, would like to hear your thoughts as well. And that is, how do you avoid overcoverage of high copy number plasmids during bacterial genome sequencing?
Yeah, honestly, we've not really found it to be a problem. When we sequence, there's enough capacity so that chromosomes and plasmids are recovered. We've never found it to be a problem where plasmids are outcompeting chromosomal DNA for coverage. I think part of it is just a technical reality, which is a lot of plasmids are supercoiled. And also, if you're doing DNA extraction, if the DNA extraction is not specifically focused on getting the plasmids out, you are probably not getting high representations even of high copy number plasmids. So yeah, we've not found it to be a problem. If you're having a problem, you can email me too, and I'm happy to connect you to a technical person.
Yeah, I haven't heard it being a problem in with our systems. I do know that there's certain, I'm going to say, motifs that give difficulty in sequencing certain plasmids, but typically if you're having trouble, you need to either contact our tech support or FAS or a sequencing specialist, and we can help you sort it out. And I just want to say that I'm thinking about more, plasmids, say, found in a bacteria that's, more native, like in soil or in air or in water, versus, it sounds like to me the high copy number are typically something that's been engineered. It's like a PUC vector or something like that. It depends on what they mean by high copy number, but I'm unaware of any in nature or any plasmids that are like super high copy number, thousands of plasmids per cell, but maybe they exist. I don't know. I've just never heard of them.
All righty, for Meredith, for the upcoming HiFi viral sequencing, you mentioned that it doesn't use amplification. Does that mean that you end up using a lot of your sequencing coverage on the patient's DNA?
Yeah, so let me clarify, we do use enrichment. It's just that we are not using a traditional amplicon-based method, for example, which is used in the Arctic method or COVID-seq or other methods which really involve taking two separate primers and amplifying those. It uses a distinct technology for enrichment, which we are holding a little bit close to the vest ahead of the product release. So stay tuned. We are anticipating having the release in the first week of November. And at that point, all the details will be out, and we think you're going to find it very exciting.
Sounds interesting. I have a follow-up question for the SARS-CoV-2 sequencing. And that is typically, you have samples and they might come in through a clinic or out in the environment, like Haley was talking about, sequencing, sewage and so forth. But what typical input material do you have, and what should we target? What kind of CT values might be the best for some of your applications so that you get full coverage, high coverage, so that you can determine the variants of concern?
So our forthcoming kit has, first I'll note that CT values and pooling of samples with highly divergent CT values is, I think it's an issue across all platforms, and getting even coverage both across the genome, but also across all the samples that are in a pooled library. So what we have found is that with our new method, we have quite robust performance for, I think, and don't hold me to this because we have our beta test site data still coming in, but we have very robust performance with complete genome coverage up to CT26, and then above that, the rates of complete genome coverage does start to attenuate. And what we have found is that, but on the positive side, because we do have that HiFi advantage and the ability to have high confidence in any one read, because it has been sequenced many, many times, we only need about 1,000 reads per sample to get complete genome coverage. And so, again, we're seeing very robust performance. A recent run that had, well, I don't want to overshare ahead of the release and get into trouble with the rest of the marketing team here. But full details will be coming at the first week of November, and we'll be very excited to share that with you all.
Thank you. Thank you so much. Haley, do you have a ballpark there? And I think there's another person having a similar question here.
So my take is you would want nanograms of material. You don't need micrograms and nanograms. And that you want, in some of the samples, the RNA is going to be degraded, and you just want to make sure that it's larger than 200 bases. And if you think it's more degraded, then you would want to use an enrichment rather than PCR. The cases with trying to PCR up fragments that are too small. I think that, and the whole, so there's the whole aspect of, I know there's all these debates about, I used to work on TACMan, there's all these debates about the CT, what CT value. And I always look at, there was some debate when people are using CT values for positive testing way up in the 30s. But, having worked on that technology and the TACMan technology and R&D, if your sample has an inhibitor, then you're not going to get close to, you never get twofold amplification. It's always around like 1.8, 1.9. But let's say there's an inhibitor and it's lower, it might take more cycles. But I would say for good coverage, you want like a CT value of 26. We've seen good data in the low 30s. But it's just like anything else, Donna. It's that, I was thinking about sewage. Crap in, crap out.
Yeah,
So,
no.
Sorry.
Don, I think that probably gets Harsh's question, you think?
Yeah, that gets harsh the question.
Yeah.
Yes.
So
Hi, Harsha.
to also weigh in a little bit more on what Harsha is asking. Like, there are some things that can be done to try to get complete genomes from higher CT values. But what we find is that those things are not, they're more of a heavy lift than what most customers are looking to do with a high throughput surveillance pipeline. Now, if you have a study where you've collected like 3,000 samples from a very specific patient study cohort, and you absolutely need to have full genomes from all of those precious samples, there are things that can be done. One of them is, with PAC Bio, the bulk of the expense for the assay is really the enrichment part. And then once you have that pool library, you can just sequence a second SMRT cell for the entire pool library, right? And with that, and then you can push some of those samples above that 1,000 reads per sample margin in that way, and get more complete genomes from that. If you really want to do the heavy lift, you can even go back and cherry pick the low CT samples off of your plate and pool those into a library and do that if you really need to get those samples sequenced, because one of the main drivers that we've seen of low performance for high CT values is not so much that the data isn't there or couldn't be collected. It's that when you have a CT value of 16 on that same plate, it's orders of magnitude more concentrated than the other samples. It just gets outcompeted. So if you are willing to go through and do that cherry picking of samples to resequence, you can get the data you need. But again, not really appropriate for a lab that's looking to sequence 3,000 samples a day.
All right, next up, do you see an increased need for decentralizing NGS in order to meet the demands of infectious disease and pathogen surveillance? Do you think, or what do you think are some of the limiting factors to this?
You want me to take that?
Sure.
Yeah, why don't you go?
I would say yes. I think it needs, and the reason why I think it needs to be decentralized is different communities are different sizes. And then in terms of shipping a sample to, from one place to another, there's the whole aspect of how quick you need to react. So, and there's been, I'd say with our platforms, we've been placing a lot of smaller units in small health departments throughout the country that you wouldn't have even imagined 2 or 3 years ago would buy a next gen sequencer. You see who's we're talking to and who's buying platforms, and to me it's I don't want to say it's shocking, but it's just amazing how far the reach of genomics has gotten at this point. And I just think it's more like any other type of supply chain. It has to do with scale. So you don't want to have a bunch of little small platforms in, say, Los Angeles or the Bay Area. But if you're out in the middle of, Nevada or Utah, Colorado, Wyoming, there's some smaller communities where a small platform would be perfect.
Yeah, I totally agree with that. And I think one of the things that the global pandemic has highlighted is that there is a utility for having a centralized database and centralized information. And, operations like Quest and LabCorp and the CDC were able to pull on those centralized networks with large capacity instruments to generate information quickly. But, outside of that, there's also an enormous need for regional solutions. Here in the US, we have a lot of viruses that are, really only of interest or of concern in, the Southwest. And so it doesn't really make sense to have one central authority prioritizing what needs to be surveilled to the exclusion of things that are of serious concern in those different regions. And I think that's part of the impetus at PAC Bio today for coming out with some more competitive pricing for specific customer types, especially public health labs, so that they can have access to the full armament of tools that have previously only been available at those larger centralized labs.
And I'd just like to chime in here and just kind of to echo what Meredith has said. And I think you have to, from the data perspective, you want centralized data storage and sharing, but you need that to be able to support decentralized instrument placement. So I think that we learned, and the last panel spoke about this, it's the sharing and the quickness of sharing and getting all that data up online that's important.
Yeah, that's really true.
Yeah, I think that ties in the next question, actually. What's your recommendation to the small clinical testing labs without strong NGS infrastructure regarding the virus surveillance? Can they contribute? So I think that may be answering that to some degree, but I'll let you guys expand if you'd like.
Yeah, I would say if there's small labs that are interested in getting involved, again, hit us up. We are ready to talk to you and see what we can do to enable your lab. And again, I think Haley was spot on in saying that anybody who can contribute data to a centralized repository has something to add to surveillance.
Yeah, and I would say absolutely. And I think it depends on what you mean by, a small lab. Or I grew up in what I consider a small town in Santa Barbara, California, there's roughly 80,000 people there. I think there's 250,000 people in the county. And the Santa Barbara County Health Department has basically they report all their cases, and they're, you know, in partnership with UCSB, and they get it all done. And, you know, it's, when I was online looking at their website, it's not that I'm like, yeah, I'm surprised that, you know, small county health labs are able to do it. And I think it's important that small health labs do it, because they understand the community in which they live, and they better understand how, I want to say, from the individuals that are either contracting the disease or spreading the disease, they understand better how to deal with them and message to them. So I think having that information in their hands in a timely fashion and going out and talking to people will help better contain it. So.
Just for some perspective, your small town is about twice as big as my county.
All right, well, we're gonna have to wrap up this session. It's already a little bit past 3. And I would like to thank both Meredith and Haley for their participation. This has been a great session. And this wraps up our second session for SFAF 2021, part 1. And we also want to thank Illumina and PAC Bio for supporting this session with the updates. And of course, many thanks to all our sponsors for supporting today's session. Next up is the poster session. So if you haven't logged into HiFi yet, please do so to engage with and look at posters, etc. Later on this evening. So we have the Tech Talks up next at 7 p.m. Eastern, 4 p.m. Pacific. And they're going to have amazing insights from New England Biolabs, NanoString, Phase Genomics, and SAS Institute. The link that you have for the sessions has been sent out, and will be available 10 minutes before we go live for the next sessions. So again, thank you, everybody. This was a great conversation today, and love to see you next time, and hope you can participate in our future meetings as well.
You know, just one thing to add real quick at the end here. If you do have other specific questions for Meredith or Haley, you can go to their specific booth through the HiFi app also, and direct questions to them. So we welcome that as well. So if you're more comfortable with that, you know, interaction, please visit, and please do so. It's a great opportunity.
Yeah. Okay, with that, we're going to wrap up our session. Thank you, everybody, for participating, and everybody that logged in to see the presentations.
Great.